Okay. So one of the questions when we look at proteins is they have a remarkable three-dimensional structure. For example, I'm showing here, a cytochrome C, four-helix bundle fold, or not big. And when the proteins adopt this three-dimensional structure, at least if they're biological proteins, often they will perform some type of biological function. In this case, it's binding a heme group. And one of the questions you might ask is how many of the features of the protein structure, as well as its capacity to bind this particular molecule, is the result of the inherent physical properties of the system without selection for anything? And how much is the result of fine-tuning through evolution? Now, obviously, in the real world, biological proteins are a convolution of two effects. And in practice, it's pretty difficult to tease out how much is evolution and how much is physics. One thing you could do is you could do a design study. You could say, okay, let me just generate a random repertoire of amino acid sequences, look at this soup that folds, pull out the ones that do, look at the structures, see if these structures, which are compact, look like real proteins, native like biological proteins, and assess whether they have a biological function. Now, this actually has been done by a whole group of investigators, including Michael Hecht at Princeton, Dan Talfik at the Weitzman, and they find that, yes, lo and behold, that these, quote, minimally protein selected for thermodynamic stability often give rise to biochemical functions. They may be a peroxidase, a hydrofolate reductase type fold, function, etc. But the question is, that's for one fold. So maybe there's something special about the How comprehensive is this in general? So how many of the observed features do we see of protein structure and function can you explain without any evolutionary selection for function whatsoever? And this is where what I like to do, computer experiments are very useful, because you are an imperfect creator. You can set the rules of the game. Now, it may be like the Roadrunner and you look down and gravity gets turned off until you look. And it's very non-physical, because as we know, gravity is always on. Or it could be something which is an interesting caricature of reality. And so what I want to tell you about today is the question of what is the balance between the fundamental physical principles of the protein and evolution. And what I'm actually showing here to get ahead of the story, including the binding pocket, are two proteins. I leave it for you to figure out which one is the biologically relevant one of cytochrome C, and another protein which was generated purely at random with no selection for function whatsoever on the computer. They have virtually the same global fold, and they even have the same binding pocket, which will accommodate a heme quite nicely. Maybe this was an accident. And so one of the questions is you could do a computer experiment, and you want to do this at very large scale, is how much of the physical properties of proteins can we see? Can we simply explain because they are bumpy objects that are compact with secondary structure? And how many of the features do we actually have to invoke selection for evolution? Okay. So that's the purpose of this talk. And then at the end, I'll show you some of the consequences of this for drug discovery. So, question? What's the least we need to put in to reproduce whole protein folds? Are there all protein folds known? How do you prove that they're known or not known, at least for single domain proteins? What's the least you can get away with? So we're going to do a series of thought experiments. Next question. We have a set of proteins. They may have a global fold, but they may or may not have the cavities, the protein-protein interaction sites, the DNA, the capacity, as well as the sequences, to actually bind, at least in silico. So they'll mimic. These are cartoons. They'll mimic the properties that you can't tell them apart. And what we're going to talk about is small molecule ligand binding sites, protein-protein interactions, and protein DNA. And so it asks, how much do you get for free? Simply if you have a library of compact structures, initially homopolypeptides that are then evolved for thermodynamic stability, in a collection of a variety of force fields, because none of the force fields are particularly good, and your hope is that if it doesn't depend on the force field, it's a robust result. If you change the force field by 2%, the results fall apart. Don't believe any word of it. So this is what we're going to do. And so let me talk about ligand binding pockets. Now, one of the things that should be bothering you is if you ever read the off-label effects of any drug, headaches, nausea, vomiting, neurological diseases, seizures, convulsions, whatever, whatever. And is it interacting? Now, people have done a really hard job trying to pick a protein target and design for it. Yet somehow you have all these effects. So there's one possibility, which is, well, that one protein you targeted does all this stuff. The other possibility is you're interacting with a whole bunch of proteins. I'll show you, in fact, you're interacting with a whole bunch of proteins. And then the question is how could it possibly be that you've designed for one that you interact with so many, when you certainly haven't done it? Or more generally, how does nature do this? So the space of ligand binding pockets. So I'm going to talk about artificial structures. These are synthetic homopolypeptides that are then decorated for stability. And native pockets. Do they look anything like real proteins? Are they statistically significant? Another thing which you kind of may guess is the fact that even though you have different global folds, okay, the fact that you have all this cross reactivity might suggest that the number of ligand binding pockets is actually quite small. Because if by accident you designed for X and you're also binding and exhibiting physiological activity for Y, one possible explanation is the space of pockets is small. Now there could be two possibilities. You could say, well, I'm actually interacting with proteins of the same global fold, and pockets of global fold are very strongly coupled, and that's the reason I'm doing it. I have the same 4-helix bundle geometry. All 4-helix bundles have a heme pocket of that shape, and that's the reason. I'm actually going to try to show you that global fold and pocket geometry are only at best weakly correlated. So you're going to find similar pockets in similar structures, dissimilar pockets in the same structure, and similar pockets and dissimilar structures, and I'm going to show you some recent work that Ambrose and Barat did in my lab, where we're actually going to pull a pocket from a completely different fold, use it to try to design drugs, and actually show results in animal division. So this is not just simply an exercise. If there is a finite number of pockets, do we know them all? If you know them all, you could then imagine, for those of you who do computational modeling, you could do threading on pocket space. It's then so all I got to do is look at a structure and say, have I seen this before? Maybe I've seen it in a completely different fold in Computational. On the other hand, if the space of pockets is astronomically large and every pocket is special, this would be a terrible thing to do. And then you got to tell me, well, why is your drug binding to, on average, probably 100 or so other protein targets? And this bears with the question of how coupled is the global fold. I mean, you may see in a lot of people when they infer function say, I have the same global structure, therefore I must have the same function. Right? Well, that's true if pocket geometry and global fold are one-to-one. But if pocket geometry, at least for ligand binding and global fold, are weakly coupled at best. And both in nature and, as you'll see in these artificial libraries, they are weakly coupled. Inferring function turns out to be not as easy as you may think. It's not just one-to-one. And as I will try to show you at the end of this talk, actually figuring out whether a pair of proteins or evolution are related, unless you have a transitive series of walks of close sequence, it turns out to be very hard. Put another way, the random background probabilities are much higher than people have appreciated for these things happening by accident. And one of the things you might imagine is one of the things you may say is, well, I got a similar global fold where I have a similar utilization of amino acid residues when there's no evolution. So it's a functional pocket where I see similar conservation of patterns of residues. So there's no relationship. And then that evolution is playing this key role. It's certainly possible. And so these are the kinds of questions we're looking at. So that's for pockets, cavities, holes. Now let's talk about protein-protein interfaces. Proteins interact. In order to have life, a minimal requirement that you're not dead, even though there's much more, is that you have interactions. If you have a whole bunch of disjoint molecules, you're dead. You may have a whole bunch of interacting molecules, you may still be dead, but it's necessary with problem solution. So what can we do? Again, we can ask question, how many protein-protein interfaces are there? Why is it that ubiquitin binds to so many things? It's got an interface, it sticks to a lot. How could this be? If the number of interfaces is relatively small, you might imagine that this happens, so there's redundancy. If every interface is special, how could this be? How coupled are the protein-protein interfaces to the backbone geometry? And this bears on how many interfaces are there, and is there any relationship between ligand-binding pockets and protein-protein interaction? Imagine I take two quasi-spherical objects and put them together. I create a little depression. I take two convex surfaces, put them together, in between them you'll have a cleft. This is a great means of regulation and control. Is there any statistical significance in real proteins and artificial proteins that ligands actually tend to co-occur with protein-protein interaction interfaces adjacent to the sites? So that might actually be the way you want to muck them up, is not necessarily inhibiting the protein-protein interaction, but focusing on the site adjacent. And if we consider a fraction of artificial proteins that know nothing about the fact that they're supposed to interact, other than they are monomeric and selected to be stable in a large variety of potentials, through docking, if the interface space is recapitulated, do you get protein-like interactions at random? So what I'm going to try to convince you of is that a lot of what I certainly thought was true was that the properties of proteins were special and highly optimized actually occur in random libraries at remarkably high probability. It's not one, but it's enough to be interesting, and this then is highly suggestive for how life could come. So, what we want to do is, the theme is I want to get rid of evolution, and I'm going to do it computationally. I'm going to throw it out. We're simply going to consider reference states with certain sets of rules and see what the consequences are. Very simple. And we're going to generate a set of protein structures and look, do they have pockets, do they have protein-protein interfaces? When I decorate them with sequences, do they look like proteins, or can I falsify the idea that they're protein-like? What do I need to do to generate selection? Now, I'm not an idiot. Obviously, functional selection is involved in optimizing. We're talking about low-level function. We're not talking about diffusion, collision, controlled enzymatic reactions. So let me consider two, in addition to the native structures, two types of protein models. Let me talk about this one first. This is a quasi-spherical protein model. What this is we take the sphere, the equivalent volume of a protein, and we fling random balls in it, fill the space, connect the path, and look, does it have what properties does this have? It doesn't have any secondary structure, as you'll see. It has local geometries, which turn out to be generated by excluded volume, and local by size comfort. And that's it. Does it have the global fold? Does it have ligand binding pockets? Does it have protein-protein interface? Quasi-spherical, like not interfaces can be linear, but that can be used to. What about the artificial structures? The only difference between these and these are again homopolypeptides, which will again be decorated, except they have, you pick at random secondary structures. They will have the positive propensity for secondary structure. They will be hyperconnected. And this is showing a little protein-protein interface on this artificial structure. This is showing a protein-protein interaction. And so these are artificial proteins. They're not selected for function at all. They're simply low-lying states in a home that are compatible and then decorated and optimized. Kind of the minimal protein. And again, we don't do anything to them. And we have different structure libraries. I won't bore you. There's the PDB structures. These are monomeric single-domain proteins up to 250 residues. There's the quasi-spherical proteins. These are the proteins which are basically this kind of compact, for those of you who know it's a Hamiltonian law kind of compact space. That's all it is. That, basically it satisfies the volume and nothing more. And how you build them is really very simple. You have a sphere whose volume is related to the number of residues as in the proteins. You fling a bunch of balls in it whose radius is the radius of a C alpha atom, an equivalent residue. Pack them. Can't have them closer than 3.8 angstroms. They can be any distance they want. Connect them by the traveling salesman problem. Minimum path, and that's my protein. Look at the properties. Very simple. And actually, coincidentally, we use the traveling salesman. It's a really good version that you can get at this Concord program at Georgia Tech for free to solve. So you simply connect the dots. I want to visit each point once to minimize the length. So travel literally travel. Okay. Artificial proteins. Basically what we do is we pull secondary structure preferences from PDB structures. You can either pull them from a distribution or pull them at random. The average structural similarity to the protein which it was pulled from is random. So the resulting global folds are not. So you can have many secondary structure arrangements for a given secondary structure pattern. And it's obviously not legal as it's. We just fold them up using our structure prediction algorithm and see what happens. And then what we do is we generate we take 20, we've actually done this up to 80, doesn't make a difference. We generate 20 random sequences using a folding potential, optimized for stability, and the average pairwise sequence identity of these guys is 70%, very low. And then we look at the properties. By the way, for looking at the cavities and the interface geometries, you don't need to definitely the sequences, because the homopolypeptide properties, because it's polyleucine or an average fine. But if you want to ask sequential questions, you can. Okay, so what happens when we play this game? What's the secondary structure concept? Let me look here. Okay, this is a very detailed version, and the reduced models don't do a spectacular job. This is the native, this is the artificial. The difference is reasonably consistent between these crystal structures, PDB structures, and artificial. What about the hydrogen bond pattern of these quasi-spherical proteins? None. So they're not hydrogen bonded. So what happens? Basically, we're now asking the question, what happens when I turn hydrogen bonding on? Now, in principle, you could do these experiments in the real lab. You could look at artificial backbones that have a couple thing. These are flexible, okay, which is actually the key. The hydrogen bonding is going to introduce a local persistence, and you're kind of packing sticks in the artificial proteins. These have a much shorter persistence, and that's the key, among other things. And the question is, if I look at the library of folds, so I have this artificial contour, it's only a protein fold and a mother protein fold as well. And you ask, what is the statistical significance? Can I find a similar structure in the library of native states of these artificial proteins and this quasi-spherical? Okay. Remember the quasi-spherical ones, you're looking at the chain contour. Now, a number of years ago, we introduced a thing called the global TM score. I mean, there's a lot of ways of comparing protein structures. The advantage of this guy is that the statistical, it goes between 0 and 1. The best average random structure alignment between a pair of proteins is 0.3. The average value is about 0.15. Okay. At a p-value of 10 to the minus 4th, as a TM score is 0.1. The advantage is it's length independent. The question is, when you look at things like the root mean square deviation, for those of you who follow this, is the statistical significance of a 50 residue alignment with an RMSD of 8 better than a 10 residue alignment with an RMSD of 2? Now, you can actually calculate it, but it's kind of hard to know. I'd have to give you the p-value. This way you can compare it. What you see is basically every fold. Every quasi-spherical protein finds a corresponding global structure, and the converse is also true. So chain contour, the geometric arrangement, when you look at it as kind of a smooth contour, is in the PDB and vice versa. Everyone is there. So the library of folds basically is doing t-test. Now, I get a little bit better. Let me put geometric fidelity, and now I got secondary structure in here. It looks a little bit closer. Everyone is in the PDB. Every PDB structure is in this random randomly generated library. The library of compact structures is done. Only if it's compact. If it's swollen, this is not true if you have multiple domain proteins or proteins. So we have a small number of structures. It's very unlikely. And actually, when people claim they solve those structures when you run this algorithm, you always find it. I can't say that there aren't any. It's highly unlikely. So, and the chain contour comes from packing, dense packing of geometric objects, because this structure is shown. You can make them a little bit better, you can make them a little bit stiffer. Okay, so you have the global fold. So there's nothing special about global folds. It's really because of a compact arrangement of atoms in a more or less ellipsoidal object. And this is kind of what you see if you look at the axiom ratios of the principal components. This is crystal structures, these are artificial structures, not bad. And random structures, well, when Michael Berlinski did this packing, he basically built spheres, and so this ratio is 1 to 1 to 1. When you look at actual proteins, most proteins kind of look like eggs. There's 3 components, okay? So the ellipsoids of revolution. There's a bigger axis, they're not spherical, actually. And that's recapitulated. Why? Because they got sticks. Hydrogen bonded secondary structure elements, that breaks the spherical symmetry. So they look like proteins. What about how the density? So if I look at the surface area per volume, the artificial and the native structures look like this. The quasi-spherical proteins are denser. They have less surface and more volume. They're better packed. This is an ideal sphere. Now, of course, when we have geometric objects where you can't violate excluded volume, you're going to have holes. So actually, if you had a perfectly packed surface volume here, when you have restrictions that they have to be sequentially connected, and they occupy a size. It is what, but it's denser than the native ones. And the native and the artificial ones have comparable density. Now this has a profound set of consequences. If I have an object which is denser, what about the holes? Let's hold it. Do a hole. That's actually what you see, and that has profound consequences as well. Native proteins, these guys have a similar distribution of holes. These guys, their holes are tiny. You don't see cavities that would bind any small organic molecules or anything besides. So quasi-spherical proteins reproduce the global fold and nothing more. You turn on hydrogen bonding, you get secondary structure between, you're basically packing bumpy sticks. The defects between the bumpy sticks are the holes. That's why the density is low. That's exactly what you see both in the real structures and in the artificial structures. You can't tell them apart. So let's talk about the protein pockets in native and artificial proteins. And this is just recapitulating. This is the average hole size, which is about 100 angstrom cubed for the median, for the average value. For native ones, for artificial ones, and random ones are tiny. It's about a third. They're tiny holes. Tiny, holes. They don't have the defects. You don't have these pockets. You can't engage in the binding. It's around a third of the size. So hydrogen bonding generated secondary structure, which generated packing defects, which generates the hole, the volume of the holes. Now the first thing you should be saying to me is, this is great, but I can have a volume which is a spherical volume, or I can have a stick of the same volume. What's the shape? I want to not compare just simply the volume. This tells me that comparing the volumes of these to the native ones is kind of pointless. Whatever the shape is, it's too tiny. But it could be here that these artificial ones have a different pocket shape, and you care about the shape, because that's dictating the kinds of molecules you can bind by steric interactions. If I have a needle and a long cylindrical hole, I can't shove, I'm going to try to shove a big fat spherical and it won't fit. So I care about this shape. So I have to compare pocket shapes. And so Mu Gao, who's in the audience, built a pocket alignment algorithm. And basically with that formula, I have two pockets and I want to compare them. So there's two ways you can compare them. There's the way in which one way is that you can say, I'm interested in a sequential alignment. Okay? If I go from the end to the C terminus, are the pockets similar? Now, of course, if you have an interaction surface, you really don't care the order in which the residues are labeled, right? You just care the shape the same. So you have to do a sequence order independent alignment. So it's like aligning a pair of sequences when you don't care about the order. You just want to see what's the maximum number of identical residues. Okay? Except here I want to compare the two geometries. I don't really care if this one comes before this one or after this one. If it's got the same shape, I want to detect it. And so Mu did a very good job building a heuristic algorithm using LSAP to actually build, and what we do is that we want to calculate the statistical significance of this. And so this is a pocket similarity score, which looks at the geometry of the pockets, and we want a score that's length independent so I can compare them, and we're going to calculate p-values. You want random. I want to know what's the probability of a set of randomly related pockets relative to random. How significant is it? I give you a number. Now these scores run between 0 and 1. 0.38, to give you the magic number to get ahead of the story, has a p-value of 0.03. So that's kind of the threshold we're using for minimal statistical significance. And this is just showing that Mu was able to build a length-independent mean pocket similarity score, and on average, it's like a PS score of 0.4 is like a p-value 10 to the minus third. This is what it's looking like. If you see one of 0.5, it's basically they're the same pockets. Okay, so try to give a feeling for how significant they are. And these are the things you got to do if you're going to play this game, because you want to ask the question, I have a set of artificial pockets, I have a set of real pockets. Is there any relationship between the two of them whatsoever, or is this an accident? How do I know if it's an accident or not? Okay, so we now have our metric of truth and beauty, thanks to Mu. And now we're going to compare. How many pockets are there? Well, it depends on the p-value. I'll come to why this curve closes in a minute. So we're going to consider comparisons of the PDB structure to the PDB structure. Artificial to artificial, which are these blue guys. The purple guys are PDB artificial. And what you see is that, first of all, at a significant value, you have on the order of about 150 to 140. Tiny number of pockets. You want to go to a p-value of 10 to the minus third, is on the order of 300 pockets. These are pockets, we're not necessarily saying they're ligand binding pockets. they tend to be a little bit smaller. They're a subset of a spatial pocket. Is less than 1,000. Every one of them, in terms of here, you can use the artificial library to find the pockets. You can use the real pocket library to find the artificial pockets. Every one has a counterpart that's statistically significant. And this is just covering the space. This is the fraction of matched pockets that the function is supported as random. And what you see is eventually, as it gets more significant, you see it's going. But at the level of statistical significance with a p-value less than 0.03, every pocket is a member of this tiny library. You can make them fancier, you can make them a little bit finer. The pocket space is small. It's not huge. So now already this is starting to give you nightmares. Like if I have similar pocket geometries, which partly reflect the stereochemistry, why don't I have a lot of promiscuous interactions? So, or maybe you do. I'm not saying the interactions are the same, because obviously there's different components. But you clearly do have them. Now let's ask the question, that's one way. So the pocket space is a small number. Is it actually complete? So what's the best match? This is random. So depending on the library, it really doesn't make a difference. This is the PDB to itself at very low sequence identity, less than 30% sequence identity. Here is the artificial to artificial, PDB to artificial. Every pocket in the PDB library finds an artificial pocket and vice versa at a highly significant. Doesn't depend on the size. This is even 50 residue pockets, huge pockets. Sometimes on protein structures, they kind of look like the surface of the Earth, or the oceans. You see these humongous pockets. But if you limit yourself to kind of pockets that are going to bind small organic molecules or alkanes, every pocket's in the PDB, every PDB pocket's there. Okay, now let's talk about pocket structural similarity for a given extent of global fold similarity. Now, if you really believe that the global fold dictated whether the pockets are the same, then when I have highly significant pockets, this has got a p-value of like 10 to the minus 6. These structures are very related to each other. These are the PDB structures. This is the cumulative fraction of representative proteins whose pocket similarity score is greater than or equal to this value. These are the ones that match. You only see about 20% of the pockets are statistically significant. Even when I have a virtually identical global fold, and I'll show you this in another case in a second. So even when I have globally similar structures, very similar structures, most pockets, in this case 78% of native ones are pockets. Now when you compare the PDB to the PDB to the artificial to artificial, what's remarkable is, and I don't know quite how to interpret it, they're more similar and more coupled to the global. Now is this a reflection that evolution is tuning for specificity? I have no idea. This is an empirical, I cannot explain this. What you do see is the fraction that are similar as a function of the global fold increases. Now, your first response is, this doesn't make any sense. But let me try to rationalize it. Let me imagine I have a protein structure, and I shave off all the side chains. Then you know that actually there's a lot of space between the proteins, and the whole thing is a pocket. It's a whole. It wouldn't be stable, but let me imagine. Now let me imagine I have a pocket, and I fill it with polytryptophan. I don't have a pocket at all. The thing is space filled. So even though I didn't change the global geometry, I can shift the pockets, I can make pockets appear and disappear, and you actually do see this during the course of what you think is evolution. Okay, now let's talk about global structural similarity, the converse thing for given pocket similarity. Let's talk about here. So this is, TM scores a 0.4 statistically significant. Okay, this is the cumulative fraction of proteins whose TM score is less than or equal to this value. So most proteins, including this is native to native, or you can pick native to artificial, doesn't really make a difference, actually are in dissimilar folds, not the same fold. Okay? TM score 0.4 are statistically significant folds. If you ask me what is the fraction of structures that have that pocket at that similarity, you can pick this one if you don't like it. There's more, but still, here we're only talking about maybe 40%. That are in structurally unrelated proteins. Most pockets that are similar are not in the same global fold, they're in a different structure. That's all that this thing says. What this is telling you is while there is a partial coupling of pocket structure and global fold, it's only weak. It's not one-to-one. So it is not true that you give me the same global structure unless they're very, 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 very similar, and I'm going to have the same pocket similarity. what you're going to see is that in this case, most of the pockets are in, globally dissimilar structures. Well, that's life. And this is again, sorry. Now let me prove this to you in the most extreme case, both in a thought experiment and in a real life experiment. Let me talk about the blue guys first. So this is a series of sequences where we freeze the backbone. Remember I told you we evolved these families? And then we look at the distribution of pocket similarity scores given a fixed global backbone, or literally is fixed. The RMS deviation of the resulting flat models is less than intensive region. And this is the pocket similarity distribution. It reflects the differences in the unit mass and the dump. And you may say, well, that's very nice. That's an interesting and irrelevant thought experiment. So now let's look at sets of proteins that are structurally extremely similar. The M squared bigger than 0.5. Very low p. Okay. That are more than 35% identical. That are less than that on the order of 35% identical and less than 90%. So exactly the same distribution. So what you're seeing is even when the global fold is the same, or very similar, you're seeing a broad distribution of pocket similarity. 0.38 is here. Most of them are similar. But some are not. These are the biggest pockets. They're moving around. So they're coupled, but not quite, is the moral of the story. There is a relationship between the global fold and the similarity. The more similar the global fold is, the more likely they are to have pocket similarity. But it's still a minority in all cases when you look at representative structures. So if you want to use global similarity to infer functional similarity, good luck. It's not going to work. Better look at what the pockets are. You need a more local descriptor. Now the next question you may ask is, and this was actually a surprising result. Let's look at high pocket similarity score here, 0.5. Remember that PR like 10 to the minus 4th? Okay? They're very similar pockets. What we're going to do is we're going to do structural alignments between the pockets and ask in native proteins, black ones, and artificial to artificial or artificial to PED and the colored ones, what is the number of conserved residues in the pocket at the same pocket position? So we're doing a structural alignment of the pockets and we're saying, okay, how many residues are conserved? And what's the frequency? You can't tell them apart. They're the same. The utilization of the native pocket sequence conservation is exactly the same as what you see in these artificial structures that don't know a damn thing about whether they're supposed to be functional or not. All they were done is that actually they were selected for a whole bunch of different structures. The dashed ones, the solid ones, these are different potentials. These are potentials without pair interactions that you can throw away any one of the potentials. You get exactly the same answer when you evolve the sequence. It's a function of geometry. Or geometry, that is. Nothing more. It's giving you this. So you can see relatively high. I mean, look, I got 5 conserved residues or 4 conserved residues in some of these things, which you'd say would be significant, right? They're supposed to be evolutionary related. Well, they're not. They have high pocket similarity, similar conservation of residues, and no evolutionary relationship whatsoever. Let's talk about protein-protein and protein-DNA complexes. How many protein-protein interfaces are there in native structures? Is the space more or less complete? We're going to ask the same kinds of questions. And if we evolve sequences, which we would pull the set which we evolved, are the interfaces sticky by accident? So when we do docking between objects that don't know they're supposed to interact, and we see at all random constellations of amino acids which give rise to conserved residue patterns. And so the first thing we're going to do is, this is how we're going to build it. So we're going to do it for the real structures and the artificial structures, the spherical structures, because we want to ask, maybe even though they don't have holes, maybe they've got interfaces. We want to again ascertain, is it the secondary structure which is giving rise to the interfaces, or is it simply that it's compact and it can violate excluded boundaries? So that's what we want to do. Now the problem, of course, is is that these are artificial structures. They haven't been synthesized. And so the first question you may ask is well, what relationship do they have at all to real native proteins? And so what Mu did is he said, I'm going to look at monomers, which are structurally similar to real native monomers complexes. And then I'm going to fling them together, dock them at random, and look at the nature of the resulting interfaces and see do we recover native-like interfaces. So then, this is a kind of restricted case. And the reason we're doing that is because we're saying, at least with respect to the geometry of the monomers in the interfaces, we know that in nature they do it. And so this is kind of the minimal test. And it's a very conservative test. Because let's imagine that I said these two things interact. They're artificial. They probably in reality don't fold anyway. Okay? And I say that they're interacting. They say, well, what relationship do they have to a known quaternary structure? And I say there isn't one in the PDB. You say that's very nice, it's a toy, it's irrelevant. So at least these have counterparts in them. And so that's the thinking behind this story. And so what we're going to do is build a quaternary structure, and since these are decorated, they have interactions. And we'll build these complexes and say, do they look like proteins or not? Again, it's the same logical idea. Have a random library, look for properties in the co-occurrence of properties in these random libraries with native. Plane of these surface patches. Native, this is the distribution for the deviation from the best fit plane in any strings of native structures, artificial structures, quasi-spherical structures. So quasi-spherical structures are shifted. You can also look at the curvature of the interface. Flat, it's got an infinite radius of curvature, so 1 over the radius of curvature is 0. If it's a ball, you try to fit the maximum ball. What you see is that, and this is the distribution of curvature, so this is perfectly flat. There's a shift away for the spherical proteins from the planar interfaces that native-like structures have. That these artificial structures more or less recapitulate. Take-home message of this very complicated slide, which is trying to show you this is the documentation you can go through. Native-like interfaces are quasi-planar, so are the artificial ones. The artificial interfaces are more curved because they're part of spherical proteins. They were packed in a sphere. Gee, we built an algorithm that looks like a sphere. Well, that's good if you know how to code, but you're basically recapitulating. We're going to now ask the question of how many of these structures, so remember, these are monomeric structures that match global folds of the monomers in a dimer. How many of them have statistically significant intervals? This is the distribution of p-values. This is a little bit more complicated. Let me think. If you look at over 12 trillion pairs and say I'm going to have at least a statistically significant p-value of an interface less than 10 to the minus 30. For the spherical proteins, depending on the metric you use, if you use a metric which depends not only on the geometry but the contact preservation, is 1 in them out of 12.5 trillion. If you do a more geometric definition without preserving the pattern of interactions, is 8. There's none, basically. The quasi-spherical proteins don't have protein-like. What about this random docking of the artificial structures? All you have to do is look at 192 million structures. This is what you need across the board. Right? There's about 50,000. So you're about 4 million times higher than what you see for quasi-spherical. Put another way, you can find native-like interfaces if you have a regular second structure. So if you have similar structures to the native folds in the library, you'll find proteins match at random. So, are these quasi-spherical proteins good candidates for the biochemistry of life? I would suggest no. While they may recapitulate the global fold, they don't have holes that are the right size, they're too tiny, and they don't have protein-protein interfaces. So at least with regards to trying to engage in intermolecular interactions with other proteins and small molecules, they can't. Now, I'm not saying that you couldn't have evolution sculpt them out and make them less. You could bore a hole in what you want. But we're talking about no selection. Okay. So absence of hydrogen bonding, or secondary structure, which is the consequence, has gotten rid of the holes and it's gotten rid of the planar interfaces, which is responsible for the intermolecular interaction. When you put it back, next question. This is the region we want. We want a negative interaction energy and a high statistical similarity to known structures. So what you do is you take the revolved sequences for stability of the monomers, you fling them together and say what fraction by accident would have sticky interfaces. This is the region. So it's got high similarity to a known interface and it's very sticky. This is the region we want. What's the fraction of these that do it? Of all the ones? It's about 10 to the minus fifth. So I would suggest that, kind of native-like, and these are native-like interactions, these are sticky interactions in these potentials. So 1 in 10 to the fifth of these guys at random would have the capacity to engage in a protein-protein interaction, which looks like a native monomeric structure and looks like a native interface, at least statistical significance, even though it has never been selected for interaction specificity, which is random constellations. Background probability is not 0. I mean, 10 to the minus fifth isn't great, but it's pretty big. Which raises the question, what's going on in cells where the concentrations are really high? How many of the interactions which are functionally competent are competing with these background quasi-transient interactions that are just simply just there because of the geometry of proteins? And I don't know how many actually do something that nature is taking advantage of, and how many are just there because they're there. They're noise. Next question. These are protein-protein interactions. What you see, if you actually look at quaternary structures, quite often is there is a ligand-binding site adjacent to where the two proteins are joined. Now, if you think about it, it could be a purely geometric effect. I have two kind of quasi-spherical surfaces. I bring them together. When I bring them together, there's this dip. This dip is where the cavities occur. Okay? Now, one question is, which is very suggestive, this is a switch, right? Suppose I pull them apart, does the cavity go away? So now I have, unlike in the case of single monomeric proteins, which always have the cavity, and you got to worry about regulating it, here I can turn it on and off simply by eliminating the protein or modulating it. I've got a means of control. Now, whether nature does this or not, I don't really know, but it's certainly suggested. So let's look at the distribution of the minimum distance of the center of mass of the pocket from the interface. It's about 4 angstroms. This is the mean. In native structures, and it's the same as the artificial dock structures. The artificial dock structures have the same kind of cavities. Like, so look at a similar geometries, similar cavities. And you see this is true. The pocket volumes are also very similar. By the way, the volume of pockets is slightly larger when you form this than the average volume of the pocket in a monomer. Next question you should be asking, which I don't have time to show you, is our pockets official possible to remain monomeric proteins? First day off, the same pocket set. So the pocket space is not only complete, you see the same pockets, so you're going to have cross reactivity, at least the potential, of stuff which is going to bind within a protein domain, as well as at these nice sticky interfaces, these sticky ligand binding sites between adjacent proteins or protein domains. You see similar things to domains. And this is just showing that. If you look at the minimum distance in domains, if you dock two domains, you see exactly the same kind of pattern. And this is just showing you where they kind of sit. So, interesting question. What happens when you rip them apart? It's a concern. No. When you rip them apart, the interfaces go away. The binding sites move and are much smaller. So if you simply do the following thought experiment, freeze the side chain geometry. This is an advantage of computation. Rip them apart and do the pocket finding. Pockets are much smaller in most of them. So you associate you got a pocket, you pull it apart, you don't. So I would suggest those of you who measure protein-protein interactions might want to do this as a means of control. And I would ask how many protein-protein interactions actually surfaces are forming ligand binding sites which are either known or unknown, and could be used as part of the biological function of the state of association. Strongly suggested. What about protein DNA interactions? This is a very complicated slide, and I'm telling you the punchline. Proteins can interact with DNA in a couple of ways, as for those of you who are not experts. One thing you can do is you can have a secondary structure, like a helix, and you shove it into a groove, right? Kind of a stick into a cavity. The other way is like binding pockets in hugs, right? So you can wrap around, you can have full circles, you can have half circles, they hug. So you can have little secondary structure elements which hug, which stick in, and those you might imagine, like a helix turn helix motif, would be trivial to reproduce in an artificial way. The more interesting ones are these concave surfaces in the artificial proteins, and a lower probability, but not zero, in the random library of artificial structures. So these are the native ones with the blue. These are the non-native ones. There's this tiny subset of guys that are not only can stick in helixes, which is not very interesting, but they can hug. Well, I mean, you're kind of starting to run out of stuff which these artificial geometric objects don't do. And so, quasi-spherical proteins, they don't have secondary, they certainly can't hug. They're convex surfaces, not concave surfaces. So they fail on all accounts. They don't engage in small molecule interactions. When they pack, they have tiny interfaces, so they're not protein-protein, might well be at best marginally stable. They don't have, the right kind of, they could form ligand-binding pockets, but that's about it. They're really so small. And they can't interact with DNA. They don't have either the concave surfaces are hugging or secondary structural elements are sticking. So they're miserable functional objects. So I would suggest that you want sticks. Hydrogen bonding generates sticks. Local secondary structural elements is these rigid little guys which are giving rise to the underlying defects in packing and quasi-planar surfaces which is giving rise to capacity or function. At least it's not inconsistent with what you see in real structure. Let me do the same exercise. So Mu did this calculation a number of years ago. We want high interfacial similarity and negative sticky structures. Evolved sequences for stability, selected for stability and isolation in the artificial library. Right? Nothing more. We then fling them and ask, do they look like known DNA binding proteins? And do they have the pattern of interactions that are in the very sticky regime? This upper left-hand corner, the regime I care about. And what's the fraction of them? Oh, about 10 to the minus third of them. So the capacity to engage in DNA binding that you couldn't tell, admittedly, in these artificial, these statistical potentials, whose geometry matches the geometry of real structures, 1 in 10 to the minus third artificial DNA monomer pairs whose geometry matches would have sequences at random. It's kind of getting, I'm kind of like repeating a story over and over again. It's the same thing. It's kind of getting harder and harder to figure out, well, obviously the probabilities in biologically optimized systems is many orders of magnitude higher. But these background probabilities, which are what we're looking at, are much higher than one might have one might have guessed, certainly I would have guessed. Okay. So the conclusions. If the world were composed of quasi-spherical random structures, I didn't have time to show you their local stereochemical quality, their phi-psi maps look like real proteins. So the local packing of dense structures generates the phi-psi maps. That's it. We then connect them, you get the global folds. So far, so good. They don't have secondary structure because they're not hydrogen bonded. They're denser packed, so they don't have cavities. They don't have the planar interfaces because they're spheres. Well, I mean, the convex, not planar surfaces. Okay, that's kind of obvious. And they don't engage in intermolecular interactions with the protein, the geometries. So quasi-spherical proteins, non-hydrogen bonded objects, are not likely to do the kind of biochemistry of light that we see. Now, they may do some other kind of chemistry with some different molecules, I have no idea. But they're certainly not recapitulating that which is observed. What about the artificial structures? They have similar secondary structure to real living proteins, but that's kind of that was pulled at random. They have a comparable hydrogen bond pattern. They have a similar shape. They have surface cavities that represent native proteins. They have every ligand binding pocket in the PDB up to 50 residues is in this artificial library and vice versa. That's statistically significant value. They kind of look like real proteins. They have the faces because they have secondary structure, which is the secondary structure elements are like sticks. That's where the planar interfaces come from, and they have protein interfaces. So they could, hydrogen bonding, if you want to conjecture, is why we see molecular function. It generates the secondary structure patterns whose defects are necessary to generate the geometry of observed molecular function. Not the sequence patterns in the chemistry, just the geometry of observed molecular function. Not the sequence patterns in the chemistry, just the geometry. So there's two features. One, you need to have the right geometry, and two, you need the right chemistry. So then you just got to decorate them with the right constellation of amino acids. What about pockets? The space of ligand binding pockets is likely to be, is probably somewhere between 400 and 1,000, depending on which metrics you want to use. And so it is very unlikely that you're going to have one molecule interacting with one pocket. These pockets are ubiquitous. They occur across folds. They occur similar folds, dissimilar pockets, dissimilar folds, similar pockets. They're weakly coupled. They have similar shapes as native proteins without any evolutionary relationship. Where they occur and the conserved residue pattern is exactly the same as evolutionarily distant proteins, or if you think of evolutionarily distant proteins. Can't tell them apart. It's kind of hard, actually. There's a bunch of horrible conclusions from this that I don't really like, but it's forced to. Similar pockets and dissimilar proteins. So you got to be careful. So if you tell me you have a pocket, you not only have to look at the same structure, you're going to look at a whole pattern of structures. It's only weakly coupled. And I'm going to show you an example of actually where Ambush made some predictions, and then we checked them experimentally to see whether these predictions bound, where the pockets were similar, but the global folds were completely different. As an example, that this is not simply a computational artifact. So if you only have a small pocket library, you would expect that drugs or small molecules to interact with a pile of proteins. Differential specificity, they're not all femtomolar binders, but enough. And what you actually see is this is the observed distribution across different proteins. Our best guess, and there's a lot of false negatives here because they haven't been tested. Our best guess is a given small molecule, a given interaction with a molecule. Now let me just show you a case of this. So this is a case where we looked at globally different, similar folds, low sequence identity. This is for a methyltransferase. This is a 1/100th of a picomolar binder. That was pulled out. A novel. This is a case for cystic fibrosis protein. These have globally dissimilar structures, about 0.35, sequence identity of 4%. Predicted structure, you pull out the pockets, you do the virtual screening. This is an animal. So this is not an artifact of some computational guy claiming. You can actually use this in practical drug screening. So you can mix and match pockets and structures to your heart's content. With respect to protein-protein interfaces, it's complete. There's actually about 1,000 distinct interfaces. There's about 30 ways a given monomeric structure could interact. There's about 2,000 statistically distinct monomers, so you wind up with 60 million monomers. We're nowhere near the space of so that's why I was very careful to say only monomeric. So let me just conclude with this. I want to establish, is there an evolutionary relationship between the proteins? Artificial and native. Similar global fold? No. Sequence threads? No. Similar binding site? No. Similar pattern of amino acid conservation, unless it's very high? No. Phylogenetic analysis? Yes. So I suspect that a lot of evolutionary relationships between distant proteins may actually not. I don't know. But for everyone you give me, I can give you counterexamples that look like it and are not, because I know. And so let me just thank my collaborators. Michael did the work on the quasi-spherical proteins. Moo is involved in all the interfacial and pocket similarity algorithms and protein-protein interfaces. Hunji has built all these magnificent structures of real, artificial, whatever. Ambush did the virtual ligand screening that I just told you about for a methyltransferase and CFTR, and Murat actually is the great experimentalist in our group who actually is validating this. So I thank you for your time.
Okay, I have 1 minute to put questions. universe of folds and pockets laid down. And it is very long because it suggests the possibility that there are two very distinct modes or eras of evolution. An early era that was dominated by physics, and then a much later era that was dominated by evolution.
So what I can tell you is I'm not sure how many folds. The fold space saturates pretty quickly. What I can tell you is if you have 25 residue polypeptides, they will have pockets that can do 80% of the chemistry of life, at least metabolic chemistry. They have pockets that match with patterns of conservation and match computational. We're actually going to check this. So I suspect that the capacity to do a lot of the chemistry in life came very early. At a crappy level. I mean, it was garbage. It was probably sub-millimolar activities. And I suspect that there were two machines. And actually, I think we have a way to get these things to spontaneously self-replicate based on physics.
So what would it have been that marked that transition into?
Once you started having ensembles of these things encapsulated, that started creating on the basis of, they probably started to build bigger things, and because of osmotic pressure burst, that's when you would start to see selection take place. So once they were encapsulated, that's when selection came. A random soup of these things would do crappy function. Was the conclusion.
It's probably not a linear environment, rather than a gradual
Yeah,
process.
I think that that's right.
It's probably what you can probably estimate with physical and evolutionary probabilities. It's pretty on. But I want to give a quick comment. I'd like to talk more about this later. I think to me, the book sounds like you are surprised by these observations. For me, finding a small probability of these molecules that looks better or equivalent to evolutionary real molecules is to me not very surprising. So that's, I think, sort of like what is the background expectations based upon what we perceive as evolutionary domain. So that's, I feel like it's a philosophical difference rather than, I mean, it's kind of unknown probabilities that we're talking about.
We have to stop there. We have a clock. Thank you very much.